Hello, everyone. My name is Rebecca Brown, and I'm an undergraduate in the Department of Microbiology, Immunology, and Molecular Genetics at UCLA. I'm currently doing undergraduate research under the supervision of Dr. Koki Morizano, and today I'm excited to share my project with you about the development of a B-cell targeting lentiviral vector. First of all, what is a lentiviral vector? A lentiviral vector is a viral vector based off of a lentivirus. There are many different types of lentiviruses, but one of the most commonly known ones is HIV-1. And our particular lentiviral vector is an HIV-1 based vector. So why do we want to use this type of vector? Well, the gene that's encapsulated within the vector, called the transgene, it's integrated into the genome of the cells that the lentiviral vector infects. So when these cells divide and proliferate, that transgene will also proliferate. This is very useful for gene therapies in which we want a prolonged transgene expression. So why do we want to target B cells? Well, B cells are a type of immune cell. And when their B cell receptor, or BCR, encounters its target antigen, B cells will divide, proliferate, and differentiate in different cell types. One of these cell types is a plasma cell. And plasma cells secrete antibodies, which have very useful antiviral purposes. They also can differentiate into memory B cells, which remain in the organism throughout the remainder of their lifetime. So when it encounters the target antigen again, it can proliferate and differentiate into plasma cells, secrete more antibodies for antiviral effects. So if we can target a lentiviral vector to a B cell, and within that lentiviral vector, we express a transgene for a B cell receptor and antibody against a target viral antigen, then once these B cells get transduced, they will express the B cell receptor against that viral antigen. Then when the B cells meet that antigen, they'll divide and proliferate and differentiate into plasma cells and secrete antibodies against that viral pathogen, which is very useful for antiviral therapy. So how do we target lentiviral vectors? Well, all viruses, including lentiviral vectors, mediate their binding and thus infectivity with their envelope proteins. So if we change the envelope proteins, we change their tropism, or the types of cells they can infect. Currently, most lentiviral vectors are pseudotyped with a protein called VSVG. Now, this protein has a very broad tropism and is very efficient at binding and transducing cells. However, this is not useful for our purposes because if we administer this in vivo, then these lentiviral vectors will bind and transduce other cell types, specifically in the liver and spleen, before they can reach the B cells, lowering the number of vectors administering our therapeutic B cell receptor and antibody. So for the solution, the Morizano lab has previously pseudotyped Synbis virus envelope proteins to lentiviral vectors. And this is important because the binding region and fusion region, the fusion region allowing for lentiviruses to infect cells, are different. So we can change what these viral vectors can bind to without changing their ability to fuse in the cells. They previously developed the construct 2.21L1L, in which the receptor binding region of the E2 protein was mutated. Thus, it has none of its original tropism, cannot bind to cells, and thus isn't very infectious and won't transduce cells. So for our project, we conjugated protein L, the PPL peptide, into this 2.21L1L construct, so it only has the binding ability of protein L. What protein L, the PPL peptide, binds to is the FAB region of antibodies and B cell receptors. Since B cell receptors are only present on B cells, then this E271 PPL will only bind to and thus only transduce B cells. And this is the cloning schematic of how we created the E271 PPL. We cleaved the 2.21L1L plasmid with restriction enzymes, inserted the protein L plasmid to create the E271 PPL plasmid. Then we checked this by digestion with more restriction enzymes, and Sanger sequencing. Once we had this plasmid, we transfected 293 T cells with another packaging plasmid and GFP as a reporter gene. And then once we created our lentiviral vector, we isolated it, purified it, and checked for the expression of the E271 PPL protein. We found that it was expressed, and thus we went on to determine its specific transduction. We used two different lentiviral vectors for this experiment, 2.21L1L, which again should not bind or transduce any types of cells, and our E271 PPL construct. We used 4 different cell types. We used the CD79-293 T cells, CD79 being a protein necessary for B cell receptors to be expressed as our negative control. And we also used 3 different cell types expressing 3 different isotypes of BCR, IgG1, IgM, and IgD. We found that the 2.21L1L pseudotype did not transduce any of these cell types, as seen by very low GFP expression, since GFP was our transgene. And for E271PPL, only the cells that expressed an isotype of B cell receptor was transduced, and specifically IgM had the highest transduction efficiency. Next, we wanted to see if our lentiviral vector could specifically transduce B cells within a population of peripheral blood mononuclear cells, or PBMCs. So first, we checked for the expression of CD19 and CD20, which are B cell markers, and we found that these CD19 positive cells also expressed IgM and IgD, which again are isotypes of B cell receptors. So since these proteins were expressed, we hypothesized that our lentiviral vector could bind and transduce B cells within this population of PBMCs. So within the PBMCs, we compared the transduction of B cells and T cells. We had untransduced cells, so negative control, a lentiviral vector pseudotype of VSVG as our positive control, because if we remember, VSVG has a very broad tropism and high transduction efficiency. 2.21L1L, which should not be transducing any cells, and our E271PPL, which should only transduce B cells. Now, the reporter gene in all these was GFP, so transduction was checked by flow cytometry. We found that untransduced cells had no GFP expression. VSV cells that were transduced with the VSVG pseudotype had high GFP expression for both B cells and T cells, and PBMCs that were transduced with 2.21L1L had low transduction of B cells and T cells, and cells transduced with E271PPL had high GFP only for B cells and not for T cells. So in conclusion, we successfully developed a lentiviral vector that specifically targeted B cells within the population of PBMCs. For the future directions, we want to test this E271PPL pseudotype lentiviral vector in a humanized mouse model to see if B cells can be specifically transduced in vivo. I'd like to thank the other members of the Morizano Lab, Koki Morizano, Carrie Antacano, Anita Wong, and Trug Pham for their mentorship and help in developing this research project. I'd also like to graciously thank the Undergraduate Research Fellows Program for funding my research, and specifically the Anna and Kenneth Taylor Foundation and the James and Meredith Henry Foundation for their scholarship. And thank you all for listening.